First, I would like to thank the organizers for inviting me to talk about this research. And second, I would like to thank you for being here in this beautiful morning. I would like to also start thanking with to extend my thanks to my research group. There's like several young people that you can see in here that are responsible for the synthesis of the molecules that I'm going to be talking about. And the overview of the talk is as follows. I'll briefly touch upon drug resistance and the challenges for drug discovery, synthesis and biological evaluations of novel monocyclic beta-lactams against Mycobacterium tuberculosis. I don't think there's going to be enough time for Staphylococcus aureus, so we're going to stick with the Mycobacterium. I'm pretty sure that you're aware of the drug resistance against different kinds of microbes and different kinds of bacteria. And it was almost unprecedented that in the 2000, some of the poultry antibiotics has been banned in the United States. Part of the problem is that we actually overuse and misuse wonderful drugs to prevent some disease and for other purposes. And that led to massive resistance against the current drugs. The resistance actually has shown up in the 80s when all over the globe reports from different kinds of microorganisms resistant to current antibiotics began to arrive. And the focus of today's talk is going to be in the Mycobacterium tuberculosis, which of course it's plagues the couple continents such as Africa and Southeast Asia. But of course it's not limited, and there's like more and more cases appear in Europe and in the United States. The problem of Mycobacterium tuberculosis is that it demonstrates itself not only with the full-blown disease, but quite a few people, millions of people on this earth is actually infected by Mycobacterium tuberculosis in its latent state. So the idea is that the Mycobacterium waits until the immune system goes down and starts to infect the organism. That's why it's usually very much related to people who have developed or are being infected with AIDS, and which brings the immune system down and allows for the full-blown disease. I cannot underline more than actually the problem of Mycobacterium tuberculosis is global, and there are several strains already known to be extremely drug resistant. And it's a daily threat. The extremely drug resistant strains, there is no cure, there is no current medicine that actually prevent the individuals from dying of Mycobacterium tuberculosis. So it's not only, of course, the most important thing is the health of the people, but it's also an additional burden to the industry to be able to treat these different kinds of resistant cases of different kinds of diseases, including Mycobacterium tuberculosis and Staphylococcus aureus, because in order to treat a patient who has already resistant bacterial infection takes considerably lot of funding. So this is the early stages in infectious diseases that actually I have taken it from a Spanish journal since the conference is in Spain. So the first understanding is that we can actually treat the bacteria this way. And unfortunately in the 21st century, even though we have electronic microscopes that they can see the Mycobacterium tuberculosis in this case, or any bacteria, we are far from actually being able to overcome that particular disease. This is a poster from the Second World War that it was being published with the hope that penicillin is actually be able to eradicate the last very big threat to humanity, tuberculosis. And I'm going to revert a little bit back to the so-called beta-lactam antibiotics, which is the core of the penicillin, and bring you back to the discovery of Dr. Fleming long time ago in his lab in University of London. And this is the Fleming's culture of Penicillium notatum, the active ingredient that he was able to isolate from a serendipitously flown penicillium into his staphylococcus culture during his vacation. It is a great effort in order to isolate the main active ingredient, and it was in 1928 Alexander Fleming named this particular active ingredient penicillium after the Penicillium notatum, isolated in 0.5 milligram per liter. It wasn't until 1945 that Dorothy Hodgkin and Barbara Roger Lowe were able to actually determine, using X-ray, the crystal structure of the compound. Penicillin belongs to the group of drugs that are called beta-lactam antibiotics. And beta-lactam antibiotics, just to refresh your memory from your earlier years, student years, are these 4-member rings, which contain a carbonyl group and a nitrogen. It's sort of a peptide bond. And they are the core in the main pharmacophore structure of the penicillins and cephalosporins. These are the 2 main antibiotics that have been produced during the Second World War in Great Britain and United States. The difference is only in the, see if I can manipulate. The difference is here in this particular chain, and it was due because it's due to the different amino acids that actually the penicillium penicillium has been fed with, which kind of like gives a little bit of a difference. Over the years, many compounds of the beta-lactam class, as you can see everywhere, the 4-member structure, have been discovered. And these are actually the main classes of antibiotics that are known to date. The penicillins, the cephalosporins, the penams, carbapenems. Carbapenems are a very interesting class from a point of view of structure, because they have, as you can see in all of the other compounds, there is just a heteroatom in this particular place, predominantly sulfur. There's carbon. And Woodward has predicted that these compounds have been synthesized first and then found in nature. And Woodward was predicted that these compounds are going to also have biological activity. For a very long time, it has been the belief was that you need to have two rings connected to each other in order to have enough strain for the bond here to be biologically active. But later on, it was discovered first by Japanese scientists, and then later on synthetically produced monobactams, which have shown that if nicely decorated, the beta-lactam structure does not need to have a second ring in order to have biological activity. This is another interesting compound. It's called beta-lactam inhibitors, and I will allude to that one in a later time. So the first and important thing is these compounds belong to the class of drugs that actually work on the bacterial cell wall. And it's a nice type of drugs because we don't have cell walls, so it's very selective to this kind of structure. Bacteria, this is a cartoon of the bacterial cell wall, consists of sugar part and peptide part. And of course, it's 2 layers of these guys. And in order to have a very strong bacterial cell wall, the different kinds of end of the peptides must cross-link. And this secures the integrity of the bacteria. And the cross-linking has been done by enzymes, which are called transpeptidases, of course, because they're going to be making the link between one of the amino acids on this side with another amino acid of this side. The blue colored structure here is the so-called leaving group. So this is a nucleophile, and this is gonna come here with the, basically using this enzyme as the stitching machine, which is gonna cross-link all these peptide appendages. So that's basically what's happening, and then the enzyme is gonna come on this end, and then this end, and after a while, you're gonna have a very strong bacterial wall with the cross-links with the peptide. So how basically the beta-lactam works, it turns out that beta-lactams were perfect in mimicking that particular link. And this is the cartoon of basically another cartoon, a little bit closer look of how transpeptidation actually occurs. It turns out that transpeptidase is serine-based enzymes, which are going to have here, this is the nucleophile, and then they'll have here a positively charged residue. And to which positively charged residue this carboxylic acid or any kind of a chargeable, negatively charged group can anchor in order to do the cross-linking. So the first is nucleophilic attack of, this is the building of the wall, and that particular upper part that I have shown you. The serine group attacks here as a nucleophile. That particular part, the D-alanine linkage, this D-alanine will gonna depart from the molecule as a leaving group, and then the bottom on the previous slide, the bottom nucleophile will gonna come here and eventually form the cross-linking. The moment the cross-link is being accomplished, the enzyme is ready to go on the next step. So beta-lactams perfectly fit in the enzyme active site, anchor to the positively charged residue, serine opens and attacks here. This is very much like the peptide bond that we saw in the previous slide of the transpeptidases, perfect 3D fit, and finally we're able, at least for a while, to block the transpeptidases. Blocking of the transpeptidases leads to diminishing strength of the bacterial cell wall and cytoplasmic burst, which eventually leads to that bacterium. What is the drug-resistant type of thing? There are two types of drug-resistant in basically, which is a natural drug resistance. The bacteria always have mechanisms to which it can use against any weapons that we can use. And also man-made drug resistance, which again comes from the fact that we used, misused, and overused the antibiotics that we have. There were such a wonder drugs that even during the Second World War, the Allies were believed by Germans to speculate with this drug and said that it just basically doesn't exist. But it turns out that beta-lactam antibiotics are very good, have a very good pharmacological profiles, very low toxicity to humans, and that's why they're called wonder drugs. One of the main, there are several different kinds of bacterial resistance, but one of the main types is production of the so-called beta-lactamases. Beta-lactamases are the enzymes that are the soldiers of the bacteria, and their sole purpose is basically to destruct our drugs, our beta-lactams. And to be effective, people have actually looked into developing a beta-lactamase inhibitors, which there's several formulations currently in the market, which they consist of penicillin or any other beta-lactam antibiotics plus inhibitors for the beta-lactamases. So what we have actually, and there are several other mechanisms that for the sake of time, I will skip. And that particular slide. So, 2 minutes, okay. So the idea is that in all of the drugs that we currently have, we have this chargeable group that it's necessary to bind to transpeptidases. Which leads me to finally what we've made. We decided to look at the fact that this particular negatively charged group is going to be very difficult to go through the so-called mycolic acids of the cell wall of Mycobacterium tuberculosis, which is very specific, very waxy, and in order to survive in our organisms. So what we have prepared is different kinds of drugs, and we have tested them, and some of them actually turns out to be a very good one. They do, however, lack that particular carboxylic acid in here. Instead, it's replaced by a different residue, and this allows for the drug to change its activity. It's no longer, it doesn't do anything to transpeptidases, it doesn't bind to them, it doesn't inhibit them, but rather actually we believe that actually goes after lipases, which are responsible for phospholipid production of the cell wall. And here in this busy table, you can see that some of the drugs that we've made, well, they're not drugs yet, they're bioactive compounds, are actually in pretty much in very good concentration when it comes down to isoniazid, which is used as a standard in Mycobacterium tuberculosis. And to wrap up the presentation, I would like to mention that our drugs are actually very good against latent tuberculosis, and they work in macrophages, which we're very pleased by that particular event. For any questions, I'm happy to answer if you have some, but for the time being, thank you very much for your attention.